welcome back. We are now going to have a Q&A, short Q&A period to follow up from the first part of your talk. And I have to say, we are being joined by people in Brazil and in Croatia, France,
hi Brazil, hi Croatia,
and Hong Kong. So
Hong Kong.
We are not alone here in upstate New York. So
awesome.
Please know that we are not. So thank you so much for that lead-in with genetics. And I have to say, it does seem like you know a lot about genetics.
It's complicated.
Okay.
There's way more that we don't know than we do know.
But you do. What even got you interested in this field?
I wanted to know what made humans think and what makes us what we are. Mm-hmm. And I thought genetics would be a simple answer, like it would just it's this, and it's just been like slowly pulling one tiny thread out of a massive quilt, and it's just starting to unravel, and now you don't know where the threads begin or end. Mm-hmm. But I'm just really, I don't think you could describe something from just its individual parts. I do think that the synthesis of things and emergent properties are really how things operate, but I can't help but, from my limited ability to understand things, the reductionist philosophy has always seemed a way for me to try and grasp complex problems and systems. So that's why I went to genetics.
Mm-hmm. And so with all this information that you're understanding the complexity of it, what do you find that people have the most difficult time grasping or maybe oversimplifying? I
think people believe that genetics is a static state. Like if you have this gene, then it's very deterministic, and that's not the case, because genes are like the blueprints for what your cells can do. And then when you turn a gene on, it's like sending the blueprints to a company that's going to do the construction. And so we can control the construction of which genes we have by the things we do. You know, if someone jogs every day and they do aerobic exercise, they're going to have many different genes on in their cells and in their muscles than people who don't do that. And people who do a lot of brain exercises or a lot of reading are going to have genes on in the regions of their brain. I mean, every time you learn something and you could remember it more than a month ago, that's gene expression that's controlling your memories. You know, when you learn something, your neurons are literally changing the way they're connected because genes are turning on that cause a neuron to grow out a little bit, then connect to another cell, then it turns on neurotransmitter receptors. So genes are very dynamic and they change all the time. And just because you have a certain genotype doesn't really guarantee that you have a particular phenotype.
And it can be affected by things like our experiences and our diet as well. Is that
Absolutely,
right?
Yeah.
Yeah. Amazing. We've had a question actually come from Brazil. And that's asking, do you see a place for molecular genetic selection in the future for dogs, particularly as it relates to sort of working dog roles, say perhaps a police detection role?
Oh, that's an awesome question. I do. I think that what we have done in the past 30,000 years of domesticating and selecting dogs into different breeds has created what I call like a color wheel or the palette that an artist has. And right now we're getting to the stage where we're getting designer dogs, which is crossing different breeds. But I can imagine that in the future, people start taking genetic diagnostics and choosing certain traits from different breeds. And if you take 5 or 6 different breeds and you select which alleles are present in these dogs and you use that informed breeding plan, you could definitely do some really cool stuff. I don't know where it'll end up. One place that's been really successful is in veterinary genomics. The incidence of certain bone disorders or like hip dysplasia or cranial cruciate problems, those have actually decreased when people actually start selecting against those traits from a phenotypic point of view. And as we start selecting genetically, I think, we can do the same. We just have to be careful because we can do a genetic bottleneck when we do that, because since genes are linked together on a chromosome, if you exclude gene A, you may exclude 50 genes up and 50 genes below A, so you may lose alleles in a population. So we have to do that cautiously.
Yeah, or end up with effects where you're not expecting them in
Right.
some other aspect of the dog. Yeah, for sure.
So that point that you bring up, so we know that genotype plus environment equals phenotype.
Yes.
So how do we, for example, with cloning, a clone is not necessarily a clone in its phenotypic expression.
I think of like the brain of a dog like a sponge. And if you have a dry sponge, you could fill it with soda, you could fill it with water, you could fill it with muddy water. So if you have cloned your sponge, that doesn't tell me what you're going to put in your sponge. And so if you had a dog that had a very traumatic, sad experience in life, and it was very fearful of people, and it would bite or act out because it was just treated poorly its whole life, if you clone that dog and it was raised in a loving environment, it might not look anything like that previous dog, but it may have mostly the same genome.
But it may look different, it may act different, it may, the behavior may not be the same. So I guess cloning is something that's quite topical and it's quite expensive, and emotionally people are so invested in their relationship with one specific dog and they'd like to perpetuate that relationship with that dog. But would you say it's probably a little bit unrealistic to expect cloning to be the answer to that?
I think cloning, I always say it's temporally displaced identical twins. So if you're someone 30 years old has a clone, it's just they have a twin brother 30 years younger than them. But I think that cloning is an emotional attempt to hang on to something that's gone. And I don't necessarily think that that's the right solution for every scenario, but I'm not opposed if someone wanted to clone a dog and had the resources to do so, but there's also the philosophical and ethical implications of bringing another dog into the world when there's dogs that don't have homes, that are in shelters. So I don't judge anyone on this. I think these are very personal decisions, and I respect people's decisions. But I just think that the context of should I clone, should I not, should just be considered in a broad context.
Yeah, and with the real understanding of the fact that you're not going to get the same animal that you
Right,
had.
because here's a way to think of it. If you had your dog, and let's say you taught your dog these 4 awesome tricks, and then you clone the dog and you just lock it in a kennel for 6 years, and then you pull it out, will it know any of the tricks your other dog knew? No.
Maybe one.
Not to go in the kennel.
That one.
That one.
I guess coming back to the idea that, genetic makeup means a predetermined outcome, it's a really common misconception that people have, almost like it's a recipe and we're going to end up with, this end outcome. Why do you think we have that misconception? Is it to do with the way the media's covered I guess, like early genetics research, or is it some other factor that's led us to kind of have this misconception?
That's a really good question. I don't know the answer. I think it's probably a mixture of a few different things. It probably has to do with the idea that we want to believe that we're in control of things, and that if I don't have the genes for depression, then I can never be depressed, or if I don't have the genes for PTSD, I can never get that. It might have to do with overly, like, as fields emerge, when genetics became big, like in the 80s and the 90s, people thought like it was one gene, one protein. That was a mantra that was in my textbooks. Then they're like, oh, this one gene can make 20,000 different proteins. Okay, that's wrong, but that's rare. That's the one exception. Oh no, 30% of the genome does. And then so what it is like when genetics first came out, our simplified models were our understanding, and I think those really resonated. And the complexities of, like I mentioned earlier, like there's microRNA that could silence a gene. People don't know about that in the general public, and there's people even in biological research labs that don't understand these subtle new emerging facts of genetics, and it is very complex. And I mean, we don't really have a good way of disseminating that information to people, because like you saw in my talk, I mean, I'm just throwing all this information as fast as I can. It's overwhelming. And I told you from the beginning, it's 20% lies. But I'm telling you, it's hard. It's a complicated field, and we don't even understand it yet.
It's interesting though in science, isn't it? Like you mentioned yourself, the more that I learn, the more I realize that we don't know much at all. And it's true. And yet the funny bit is that the people who don't know anything at all think that they have all the answers. I'm
going to give credit to my high school physics teacher, Mrs. Cook. She used to say, you have to know enough to know that you don't know.
And it's actually a psychological phenomenon called the Dunning-Kruger effect.
Yes,
it's been researched.
Oh,
it's a thing. But it's also hilarious. Like the fact that to realize what you don't know, you need to know enough to know that you don't know.
Right. So I think it comes from the idea, like, if you don't study anything, I'm just saying anything. I'm going to say genetics, but it could be cards or it could be quilt making, whatever. If you don't do that, in your mind, you're like, oh, I make a quilt. I just cut this out and I stole that.
Yeah.
But there's all these subtleties involved, and the subtleties are lost if it's not your art. And it's not a criticism of anyone. It's just, whatever it is you don't do, in your mind, you're like, oh, I know that's just this.
That.
Yeah.
But it's interesting in science, isn't it? The more that you do research an area or a topic, you kind of have to deal with that discomfort or make peace with the fact that there's still so much
Yes.
to learn and that we are just contributing our little pieces of the puzzle.
It makes me feel good
great
because I didn't want to take
understanding.
anything away from the next generation of scientists. So
Of course not.
I've left a lot of stones unturned for you guys.
I came to answer a question and I've left you 10,000.
There you go. Yeah.
So one of the areas where we do feel this sense of knowledge is, like you said, that visual breed identification, where we are not very good at it, mapping the visual to the actual DNA. Can you tell us a bit more about that? The
Sure.
whole study that you did with Dr. Voigt?
Yeah. We looked at a number of dogs that were adopted at shelters, and we had pictures of them and video, and we had people guess what breeds they were, and then we had DNA tests done, and then we tried to see like how much overlap there was, and there was very little overlap. And then we did a follow-up study where we asked hundreds of people who are experts in canine or animal, like dog handlers, trainers, veterinarians, shelter workers, and out of hundreds of people, even when 300 people are saying that's a lab mix, it may not be. And I understand, I have a handle on some of it. There's a paper that came out a while back. Adam Boque in 2010 had a paper called Simple Genetic Architecture Underlies Morphological Variation in Dogs. And that paper said there are about 40 regions of the genome that control what we consider to be breed-defining traits. And that's like, floppy ears or erect ears, long fur, short, the size of the body, the short
Snout
skull.
length, yep.
So what happens is that, if there's 25,000 genes in a genome and 50 of them have these breed-defining anatomical features, you could have 11,000 genes from a Great Dane and none of the 50 that have the Great Dane feature, and you'll never, if it's the size of a Chihuahua, you'll think it's not a Great Dane, but the gene that controls size in dogs, like IGF-1, is one gene, and if you didn't get that from the parent, then you're still the offspring of the parent, you just don't have the size of the parent. And that's like when I showed you like how you get half of your genome from each parent. If you don't get some of those defining markers, then you won't have the features. And I'd just like to add one other thing that one big misconception people have about breed and genetics is that people think that breeds are clones. Because to the layperson that's not like a fan of a particular breed, all Pekingese look identical. And in humans, when people look identical, we know that they share DNA, that those are identical twins, and we're like, they have the same DNA. But we've selected German Shepherds to have the same coat color patterns and the same skull shape. And I think of it like if you imagine the genome as a rope that's like stretches, a mile, and you had 50 pieces of duct tape, and you could just wrap 50 pieces around duct tape in a mile. Those are the places that give us the features of the dog that are fixed in a breed, and the rest of that mile of the genome can be variable between the members of the breed. So they share, what they share in similarity isn't everything, but people think that. I think that contributes to that, those misconceptions about being able to guess breed as well.
Yeah, fantastic. Okay, well, we are going to take another really short break and then come back with the second part of your presentation. We want to remind everyone watching that they can join in on Twitter, Facebook, or Instagram using the hashtag SPARCS, SPARCS18, SPARCS18. And we look forward to hearing more from you soon, Chris.
Thank you so much.
Sounds great. Can we request pictures of puppies and pictures of older dogs too?
Yes, please.
Pictures of puppies reading?
And cats typing.
Cats typing, hamsters too. Hamsters are cool.
Hamsters are good. Okay.
All right, thank you.